Right, this is a result of possibly one of the more straightforward pieces of electrophoresis that you may already be familiar with. The one I put together a little bit earlier is different dyes, and you can see if we're right close in, well, we can hear more if you're looking directly down, can't we? That there's okay. You can pick out the different patterns, and you could equate them to being either the crime scene suspect or whatever you want to do. But it illustrates the point quite nicely, and it's low faffing really. There's no staining. There's no particular hazard associated. But let's talk now about the DNA one. We can see some colours, but we better not get too in front of ourselves because it isn't DNA, is it?
That's right. All the colours you can see here are simply the loading dyes, and that basically shows that the electrophoresis has worked.
Right, yeah.
But the DNA samples are not visible. And so what we're going to try and do is we're going to try to stain the DNA.
Okay, so let me just move this out of the way then.
Okay. Now there are various types of stain that you can use. These are safe stains that wouldn't be causing you any mutagenicity problems. Okay. We're going to try one of these, which is this one.
Because some stains have got a little bit of a bad press, haven't they, in terms of whether they should be being used or not.
That's right. They're trying to stain the DNA.
Yeah.
And to stain the DNA, the best way is actually to go in between the two strands of DNA and attach there. But there's a possibility that could cause mutation in your body cells if you're exposed to it. So we don't use those sort of stains in schools.
For sure, yeah, okay.
So we're using this stain which doesn't go in between the two strands, it just coats the outside of the DNA.
Yeah.
It's much safer, but it doesn't tend to be as sensitive. So we're just going to pour the stain on the gel, and I'm going to leave it there for 5 minutes. And then I will I'll take the stain off and I'll wash off the gel and leave it standing for the stain to wash off the actual gel completely, and we then should be able to see the DNA.
The bands of the DNA,
if it stains them.
Okay, right, so we'll come back to that in 5 minutes or so.
Come back to that longer, yeah.
Yeah.
It might be a bit longer than 5 minutes.